Hello and welcome to Nikolay's genetics lessons, and today I have prepared two multiple choice questions for you. And the first question is, what is the only level of protein structure that does not involve covalent bonding? And here is the four variants to choose from. Let me show you this picture on which we see four levels of structural organization of the proteins. The most complex would be quaternary structure. In this level, we see that many different polypeptides can fold and then can arrange into the complex structure, but each polypeptide here can be separate protein, but when they aggregate, they usually aggregate due to hydrophobic interactions, but also may have a covalent bonding in a form of disulfide bridges. This protein here is hemoglobin between 4 subunits. There is no disulfide bridges, and what you see here that looks like bridge is actually heme in the hemoglobin, which contains iron, which bind oxygen. But let's pretend that here we can also find disulfide bridges, which is the case in many other variants of the complex tertiary structure, where we can find that proteins, subunits can be bound by disulfide bridges, and disulfide bridges is covalent bond between R groups of the cysteine amino acid. So we can show it as sulfur binds to sulfur, and in reduced form it is sulfur and hydrogen and sulfur and hydrogen, and when oxidized would form the sulfide bridge. The same disulfide bridges we also can find in tertiary structure. Again, not necessary, but we can find in certain proteins. So disulfide bridges participate both in tertiary structure and quaternary structure, shaping these proteins, giving them this three-dimensional form. This is, of course, is not going to be the only force that helps to fold proteins. There also can be hydrophobic hydrophilic interactions. There also can be ionic bridges, van der Waals forces, and also hydrogen bonds. Let me skip secondary structure for now, but in primary structure we basically can see sequence of the amino acids which are connected through peptide bonds, which is covalent bond, type of the covalent bond. And the only variant where covalent bonding is not participating in forming structures is going to be secondary structure. Here is another picture that shows in much more detail, and you see that secondary structure is going to be alpha helix and beta pleated sheets, and beta pleated sheets are formed due to hydrogen bonds, and the same we can see in the alpha helixes. They are also formed due to hydrogen bonding. Now we can give an answer to the first question, what is the only level of protein structure that doesn't involve covalent bonding, and it is secondary structure. And now we can move to the next question, how many amino acids per turn in alpha helix secondary structure. Alpha helixes can be of the different types, can be right turn or left turn, or we also say clockwise and counterclockwise. In the right turn alpha helix we are going to find 3.6 amino acids per one turn. Not much to explain here, you just have to memorize this number. It can be useful to know this number because you probably would need to know it in order to solve many other problems on your exam. And this is all for today. Subscribe and see you in the next video. Goodbye.